Hello, I'm going to talk to you today about vitrification. This is a very important topic in cryobiology where we're trying to actually preserve biological systems, living biological systems in a glass without crystallization. So we've talked in other lectures now about what happens in the presence of crystals. In this lecture, I want to talk about how we can try to avoid crystals and still preserve biological systems. So you see on the left the typical inverted U curve that we've talked about before in other systems, and you still see the far left. You have the inverted U curve, and that's happening because of the presence of large crystals and basically salts that are being kicked out by those crystals that are concentrating around proteins and membranes and causing what we call solution effects or injury from dehydration. In the case of the faster rates that you see over on the right, you have intracellular ice formation, which we talked about before is generally considered a lethal event, except for very specialized cases in attached cells and cells that are attached to one another. But then you see an interesting effect here with these Saccharomyces cerevisiae, this yeast, which is that the viability comes back up as the cooling rate continues to increase. So you have cooling rate on the X, viability on the Y, and you have that inverted U that we've talked about, and now you have this new leg, if you will, that's turning it into some kind of a sigmoidal curve. And that is really interesting, and that's kind of what we want to talk about here. So if you get to these really, really fast rates, so here's what we talked about before. Ice forms in slow cooling, it concentrates solutes around the cells, it dehydrates the cell, and there's injury associated with that. In faster rates, you have ice that forms, it forms around the cells, but then it also forms inside the cells and can be injurious and kills cells that way. What this is showing is that there's this intermediate, there's another route basically. As you go to really super fast rates, you can avoid these large ice crystals entirely. And if you're doing this in the presence of what's called a cryoprotective agent, which is a chemical, like an antifreeze chemical, biologically non-toxic, hopefully, you can actually get really high survival. So this is from the work of Dumont, and this concept that we would become a glass instead of a crystal, that's really the heart of this whole vitrification concept. So it's been used in nature. First off, we know hibernation is not in the presence of ice, but that's what bears and other mammals will use to survive the winter, under the snow usually, in a den. There are also animals that can survive in the presence of ice. So there's an Antarctic cod that swims around with antifreeze glycoproteins in the blood that basically attach to ice like an antibody. So the ice is the antigen to their system, and then they have a glycoprotein or an antibody to that, if you will, that keeps it from growing. We also have the Rana sylvatica, which is a wood frog that can survive with up to 65% of its total body water in the ice phase, but they confine it to the vascular system in a very special way so that it's not injurious to the animal, and the animal can eventually thaw and basically hop off into the wilderness again, and it can do that repeatedly during the winter. And then you have gallflies and periwinkles and garter snakes that also can survive a certain amount of ice in their system. If you look, there are also these other systems, that's a tardigrade or a water bear, Artemia, brine shrimp, and even Antarctic nematodes, which they produce their own cryoprotective agents. And in some sense, the Rana sylvatica and these others are also producing their own chemicals, but these over on the right are producing much larger concentrations typically, and they are able to adapt themselves and essentially go to a glass rather than a crystalline phase. And once they're there, they are very stable for extended periods of time. And so this is, what we want to learn from nature is how can we do that in cells and tissues for biomedical purposes. And so this has actually been done in a number of systems that we're going to talk about. Probably most impressive and largest is in an organ. So this has been done by Greg Fahey even back in the 1980s. He's continuing to do innovative work in this area. And what you see on the right is a kidney that was infused with basically a cryoprotective agent or an antifreeze. It's biologically non-toxic, and he was able then to cool it down and create a glass. And then you see a failed kidney on the left where crystals formed. If you so you're trying to reach this glass instead of a crystalline form, you can fail by either crystallizing. Usually on warming is where the failures occur, or you can even crack if you have differential warming from the edge to the inside of the organ. So this is a topic for another day, how to rewarm and how to cool a whole organ. But the concept that you can vitrify is really what we want to talk about here. How do you do that? It's also been applied in veins, and this is just showing you an example in a vein. This is a work of Song et al. In Nature Biotechnology, where here is a slow frozen vein. You can see these large white areas. Those are actually ice crystals. And if you are using the right cryoprotective agent and the right cooling rates, you can see that you don't have those large ice crystals. This is by a technique called freeze substitution, where you can actually look after infiltrating the tissue with a resin and cutting and staining, you can see where the ice was. And then this is some higher magnification of those veins. And then on the left, you can see frozen versus vitrified behavior pre and post vitrification or freezing. And you can see that you're largely, after vitrification, you're largely the same in terms of functionality of the vein versus in the frozen where you have a large drop. So you have viability and function in the vein, which was a really big deal. And of course, that's one of the reasons why it made it into Nature. How do we see it? Well, you can see here in the bottom left, you can see through something like VS55, which is the common cryoprotective agent. When it's vitrified, you can kind of see through it. But if you start adding things like nanoparticles, or you're adding, over here, we have a big piece of liver. These things are opaque, so we need other ways of looking to see whether we've reached a vitrified state. And this is a calibration curve in both solution as well as this particular one is in tissue, but we've done it also in solutions. And you can see that the concentration for the glycerol, in this case, the cryoprotective agent or antifreeze that we're adding, actually leads to a higher X-ray attenuation or Hounsfield unit. And you can see this jump here around 6 to 8 molar. That's where you're transitioning from a crystal to a glass. So we know at these lower temperatures that you're going to have this transition, and we know what that Hounsfield unit means then in terms of being vitrified or crystalline. And we can see that over here in terms of looking at liver that's being loaded with 6 to 8 molar glycerol over a long period of time. And then what happens when you put it into liquid nitrogen. You can see, at the highest concentrations, the whole thing here is vitrifying. And here's your scale for the high concentration. And, over here, you can see that's the highest Hounsfield unit that we have on the calibration. So it's certainly vitrified. And then here, this was given less time and less concentration. Only the edge of it is vitrified and so on. And pretty soon you have a competition between crystallized fraction in the center and vitrified on the edge. And then these are very likely just crystallized. So this is the type of thing that you can use to assess vitrification in different systems. You can also see whether you're cracked or crystallized. These things will show up with micro-CT. You can also use differential thermal analysis, or DSC, differential scanning calorimetry. In this case, you're looking for a crystallization event, in this case, a latent heat of melting, where you can see that in a sample versus a reference container. And you're basically, you're able to see this reference pan is doing nothing, but there's a large exotherm that is coming, well, in this case, it's an endotherm because of the melt. If you were going down, it would be an exotherm. And so that difference tells you that there's a phase change. In the case of scanning calorimetry, you do power compensation. And so you're either absorbing or compensating by putting energy in during a phase change event. And you can look at the amount of heat that's going in or out of the sample to tell you that there's a phase change event. Both of these are useful. DSC is the one that tends to get used the most because it's more it's more quantifiable, more robust, at least in smaller systems. And so what does this look like under the microscope? Well, you can see this is an NaCl water or salt water solution, brine basically. So there's ice crystals that are growing. You can go to lower and lower temperatures, and then eventually you even see what are called eutectics or secondary solids that show up in part D there. And if we look at the corresponding DSC, we have a freezing event. So this is a large exotherm due to ice crystals forming in the solution. And then as you drop down, you have a supercooled eutectic formation that's down below -40. That's because eutectics usually are up around -21.2 or so. And then on thawing, what you see is the eutectic is actually lining up much more with that -21 that's expected. And the ice melting is around 0. And because it's not a pure solution, but NaCl water or binary, you see that that actually drags over a significant, from -20 all the way to 0. And you can see that over here where we're looking at that behavior on thawing with different concentrations, 1, 5, and 10x at NaCl, and even a eutectic mixture, which is quite concentrated. And you can see what the endothermic or heat flow up would be. And you're comparing between the eutectic melt and then the water melt. And as the eutectic is small at 1x, the water is large. But as you start moving to higher and higher concentrations, you can see eventually the eutectic is the largest, and then there is no ice melt. And we can use that information to actually help construct whole phase diagrams so that you line it up with the temperature where you're seeing the melt, and you're lining it up with where you see the eutectic. And if you do that enough, you basically begin to piece together a whole phase diagram. So that allows us then to also do calculations based on the more NaCl you add, can you calculate out where the heat release is, and is it due to eutectic or is it due to water? You can see the water heat release here is definitely concentration dependent, and the eutectic is also here going to be concentration dependent. And we can then normalize it in a variety of different ways. But this is just showing us one way to use that phase diagram and DSC data. Here's lots of different phase diagrams that have been published in the literature, and you see some early in the 60s, which is very interesting. And you can see dimethyl sulfoxide, which is a cryoprotective agent that people still talk about and use today. Some people consider it now a neurotoxin, so there's a lot of work at the NIH and in cell therapies now to try and find alternatives to dimethyl sulfoxide. But back in the 60s, when all of this was very new and exciting, there was a lot of interest in the use of DMSO to try to cryopreserve lots of different cellular and biological systems. And so that's why it made it to such a big journal. Over the years, there were many other CPAs that people found, glycerol, as well as trehalose and sugars and so on. And so you can find a variety of material out there in cryobiology, biophysical journal, and so on. Moving from a binary phase diagram, which doesn't really have a glass in it, so the salt water doesn't really show glass behavior, Once you start adding these other cryoprotectants that are alcohols and sugars, we should begin to see the phase, what we call supplemental phase behavior, where there are going to be some glass transitions. And so this was the beginning of that, that we're showing here for glycerol. You see some of the first, this eutectic behavior, very similar. The latent heat release is very similar. And we can do that for both raffinose and glycerol. People then have done this for glycerol water and trehalose water. And you can basically see where the glass transition curve is for these solutions. And this is sort of a flipped one, if you will. Sometimes you'll see these phase diagrams flipped a slightly different way. But this is what we've been calling, like the melt is here, and then this is the glass transition curve. And so if you look over here, the melt is here, and the glass transition is down here. The devitrification peak occurs on rewarming. And to kind of orient you, Greg Fahey put something, very nice, highly cited article in Cryobiology that I encourage you all to take a peek at. There's a lot of information in there. But the basic way to look at these phase diagrams is if you aren't at a high enough concentration, so, you're somewhere below 20% here, which is going to be in the 3 to 4 molar-ish range usually, and you're coming below the melt and you're not traveling rapidly enough, you're going to have a heterogeneous nucleation event, or you're going to have a homogeneous nucleation event, which is what this T sub H means. And you're never going to make it to the glass without crystals. So what Greg did is he said, well, let's go to a higher concentration where, the melt begins to come down, the homogeneous nucleation begins to come down, and I'm getting these melt and TH closer to the TG. And if I can get close enough, then I can traverse that window where crystallization events fast enough that I'm not going to damage my system. That is then countered by, as you go up above 60%, you're going to have some, potential toxicities, there's so much concentration of CPA in there that you're really, basically getting beyond what the biological system can take. And so Greg basically argued that, you should try to be in the third zone here, so you can reduce the distance between the melt and the glass. You should try to get there, as cool and thaw as fast as you can. And there's also some other concepts around increasing pressure, which we'll talk about a little bit later. That's an isochoric vitrification. That's an idea that Boris Rubinsky has been championing, but that also was something that Greg talks about in his article here too as well. So what you're really trying to do is avoid crystal growth and nucleation. If we travel down from the liquidus, which is sort of like this is where you're a liquid over here, and you're going down in temperature. As you go below the liquidus, you're supercooled. As soon as you're supercooled, that drives crystal growth, but you need a nucleus. So what is interesting is that the nucleation rate at these high sub-zero temperatures is not super high. But once you have a nucleus, the growth rate of the crystals are huge. And so what you'll see sometimes in these highly concentrated CPAs is that you get a lot of nuclei, but they haven't grown out because they're happening at a very low temperature. Or in the case of, like we talked about, intracellular ice formation, you may have a single nucleus in a cell, and that may be enough to crystallize the entire cell. So these are just sort of things to think about. A cell may have a single nucleus that goes to a single crystal that goes out throughout, or CPAs may have multiple crystals, and they just don't have the ability to grow because diffusion is so slow at these low temperatures, and you can see the crystal growth rate drops. So that's something to keep an eye on. So it looks a little bit like this. If you have a pre-nucleated, this is a 6 molar PBS solution, you can crystallize out much of your solution, and you have a little bit of glass transition there. You're coming back, you have a glass transition, and then you have a melt. If you do not crystallize, nucleate your solution to start with, you're going to supercool, and you'll basically have a nucleation event at a much lower temperature, and it will be incomplete. So rather than having fully nucleated, you're going to have a partially nucleated solution. As you come back, you're going to pass through the glass transition temperature, and then you get a massive devitrification peak. So if you see here, this liquid here is supercooled, and it basically then all turns into crystals as you devitrify and then you melt. So the melt is going to be pretty much the same, but the way those other and the glass transition will be similar, but then you're going to have your exotherm here due to latent heat is going to be quite varied depending on whether, where you have nucleation and crystal growth. So this is just some other work that was done by Hay and McFarlane back in the 90s, and they were doing this with an optical DSC. So you can see, like I said, you get to these lower temperatures and higher concentrated CPA solutions, and you're getting these spherulites, or basically lots and lots of nuclei that are just growing independently. And you can study them and see how fast they're going and what their crystal growth rates are. It's very important to understand. And again, they're using DSC to do that type of analysis. Some of the more salient work that was done in the field, Rasmussen, Franks, Toner, this has been reviewed in a variety of works. Looking at the kinetics of crystal growth in these CPA solutions, lots of different folks have looked at that. And then, phase relaxation and recrystallization also has been a huge topic. This is really just for your information, and you don't need to go further with that if it's not of particular interest. So vitrification solutions have been studied extensively. They involve, alcohols, sugars, and polymers. PVP is a polymer. And, Greg himself has, I don't know how many different vitrification solutions that he's worked with, but they tend to be in this, 8 to 9 plus molar concentration regime. And this 2004 article was a review of some of those solutions that he worked on. One way to look at these crystal growth rates, like I showed you, is time temperature transformation curves. And what it's basically saying is temperatures on the y-axis, time for the transformation on the x-axis. And you have a period of time, if the time is low enough, where you can get past this crystallization nose and get to the glass. And that's really what we've been talking about, is doing that. If you don't travel fast enough, then you're in the crystallization regime, and you will form a crystal. So this concept of TTT curves has been around for a long time, not just for cryobiology, and is just another way of looking at it. A smaller, or a different way of looking at it is critical cooling and warming rates. And what you're looking at here is basically the use of DSC in these different, so you're you're more and more concentrated as you go down, and you can see the rates that you're using to get to the point where you don't have crystallization. So you need to get to really rapid rates with 40% dimethyl sulfoxide to have less and less Q, Q being crystallization or latent heat that you're seeing on the way down in the solution. At 45% dimethyl sulfoxide, you're able to basically, after 10 C per minute, you really don't see much at all. It's more like 20 C per minute. So there was Boutron came up with an approach. He came up with an argument here, and even an estimate based on basically 0.2% ice is sort of his definition of what is vitrified. And then you can do this for whatever your CPA concentration and type is. You're going to have to get your K4, which is a constant for each CPA, and the Q max you're going to have to measure for your particular CPA and its particular concentration. But then you can do this this calculation. Similarly, for warming rates, what has been found is that you're going to have a melt, as we've shown with DSC. You have a devitrification piece, which again can be measured with DSC, And so if you have those two, then you can actually calculate out the critical warming rate. And this is the warming rate below, it's about 0.2 to 0.5% ice formation in your solution. And so this is just a different, again, another approach where A and B are constants for a given CPA at a given concentration, and there is some DSC data analysis needed. But these are the approaches that Boutron and others have used in the literature to get critical cooling and critical warming rates. They're important because they're telling you, basically I can cool at a rate fast enough to avoid ice on the way down, and then I can warm at a fast enough rate to avoid on the way back up. And you need to worry about it for both cases. And they're actually linked. So if you look, this is actually DP6, VS55, and M22 going up in concentration. These are very common cryoprotective agents. And this again is, M22, VS55, and DP6. And this was from a review that Zhonghou Han just published in Cryoletters this last year. And you can see that the critical cooling rate, or the rate needed to achieve a glass, basically drops with concentration. And that's what you would hope. And that's very similar to what we've been talking about already. But then what's really interesting is that the critical cooling rate is linked to the critical warming rate. And what you can see here is that, pick a rate, in here. So you're going at, let's say, 1 C per minute. Well, okay, pick this one, 10 C per minute. And now you're basically having to travel between, 100 to 1,000 C per minute in order to avoid crystals on the way back up. So you're usually traveling 1 to 2 orders of magnitude faster on the way back up because you pre-nucleated your system on the way down, and they want to crystallize. Like those curves I showed you, the crystallization at higher temperatures, those rates go up. And so if you've got a nucleus in your solution, it's going to want to grow. Okay, so this is just looking at some of the literature. You can dig into this if you like. Notice that they're at low concentrations. There's not a lot of data, and that's because these are super high rates and very hard to look at. So there's some new techniques in our lab. Joe Congas has been working on with laser calorimetry that I'm hoping he's going to put into one of these lectures soon as well. And these are some of the techniques that actually were used to put some of those data points on those curves that I just showed you. So there's DSC, there's plunge cooling and warming, where you're actually doing post-mortem assessment, say, of a droplet to see, can I see through it? Can I do XRD and see that there's no crystallization? There's also fast scanning calorimetry, but you need to be using a very special instrument for that. And then laser calorimetry is what Joe Congas and others have been working on. And then I'll stop here. This is our final slide. This is something we just published. It's coming out this year. And this is why we do it. Once we understand everything about what these cryoprotective agents do and how they can help you, get to a glassy phase, we can then do what Greg Fahey did a long time ago and perfuse them into kidneys. And then we can cool them down at rates sufficient to actually hit the glass. And here we're showing we can do that. And the interesting part about the work that we did here, much of this is just a repeat, if you will, of what Greg did. But now what we've done is we've actually added nanoparticles in this particular piece of work and shown that we can actually bring them back from this vitrified state. So we're showing we can perfuse this kidney full of CPA. We can show by micro-CT that it's vitrified. And in another lecture, I'll show you how we can use that information along with our ability to perfuse in iron oxide nanoparticles. We can rewarm them in a radio frequency field and bring the whole organ back. And we're working towards getting that organ to be transplantable. The viability is as good as what we see with just the CPA going in and out. So in summary, natural systems use cryoprotective agents to survive freezing or vitrify. I gave you some examples of that with that nature-inspired slide. Vitrification is the avoidance of ice, to achieve a glassy state at low temperature. And vitrification and rewarming have been successfully preserved, used with cells and thin tissues. Organs have been vitrified, like Greg Fahey and recently us, but never successfully warmed yet for transplant. And we're still working on that too, just to be clear. Vitrification can be studied with various tools, DSC, microscopy, computer tomography, and others. Phase diagrams identify melt, glass, devitrification temperatures, and solution as a function of cryoprotective agent concentration and type. And the nucleation and crystallization are separate phenomena, both driven by supercooling. And CCR and CWR, those critical cooling rate, critical warming rates, are rates that practically avoid ice on cooling and warming respectively. These have been quantified in numerous CPA solutions at high CPA, and I gave you a review so you can look up and go back to the original articles if you'd like, or just use the review. And low CPA solution assessments remain a challenge for faster calorimetry. So we're working on new ways to look at this for droplet systems, which is that faster laser calorimetry. And rodent organs can be vitrified, and uniform heat generation approaches may help, and that's what our nanowarming program is about. And I've left you some questions here you can ponder, and we can discuss if you'd like, to help you kind of dig into the material. And with that, I will stop, and thank you for your attention.